Science Communication Specialist at the IGI, Innovative Genomics Institute of UC Berkeley and UC San Francisco. Kevin earned a bachelor's degree in biophysics at Johns Hopkins University and a doctorate in biophysics at the UC Berkeley lab of Professor Jennifer Doudna, co-discoverer of today's subject, CRISPR, which Time magazine called one of the most significant discoveries in the history of biology. Dr. Doxzen's title, Science Communication Specialist, means a lot to me and to other Bay Area Skeptics board members, especially coming from Jennifer Doudna's lab. After all, Dr. Doudna received Wonderfest's Carl Sagan Prize for Science Popularization just last year. She is a fine science communicator herself, but it is Kevin who speaks for the IGI and often for Doudna, to fellow scientists and to the public. We are in for a great science communication treat. Please welcome Dr. Kevin Doxzen.
Well, thank you for the kind introduction. My name's Kevin, and I did my PhD in the Doudna lab, which was really exciting. And so I started in the lab around the end of 2012, and the first CRISPR paper came out of the lab in 2012. So I got to see, through my 5 years in the lab, see the evolution from this technology from what I'm going to talk about today, the discovery, to where it is now. In well over 50 countries, already made a CRISPR baby, which we'll talk about at the end of the talk tonight. So while you're listening, since this is a SkepTalk, I want a lot of interruptions, kind of like, I don't really agree with that, I don't know what you're talking about, so just please interrupt me throughout the whole talk. And whoever has the best question at the end, you'll get some CRISPR playing cards. So we have that. We also have some temporary tattoos up here, so I really want an engaging talk. So CRISPR, who knows what CRISPR stands for? Oh, we got one. What is it?
Clustered, regular, interspatial, short, palindromic, repeats.
That's, 98%, clustered, regularly interspaced, short palindromic repeats, but you got it. That was really impressive. So when people think of CRISPR, a lot of them usually think of just one protein. And I'll talk about what is CRISPR as it's a system, but usually people, when they hear the word CRISPR, it's actually just a protein. So I also have a 3D printed model up here, but this is CRISPR, this green guy. And so CRISPR is a tool that people are using now to edit DNA. And so people have wanted to do this for a very, very, very long time. It's a great way to answer any biological question, is to turn off the DNA. So there's some people at Berkeley who are really interested in how do butterflies get the colors on their wing. The best way to solve that question, let's go into the DNA and turn off genes one by one by one until we start seeing the color on the butterfly wings change. And so there's really cool papers. We can see them repainting the wings of butterflies using CRISPR. And so this tool has opened up all new questions in biology. And so also feel free to record, take photos, whatever, of this presentation. So what's this?
Bacteria.
Bacteria? Bacteria, right. And so bacteria is everywhere. You are as much bacteria as you are human. Right, so there's about a 1-to-1 ratio of bacterial cells on and in your body as there are human cells.
Numerically, right? Not mass?
Numerically, not mass, because you don't look like one giant bacteria because they're really, really small. Orders of magnitude smaller than a human cell, but 1-to-1. So bacteria exist everywhere. There's some really cool studies done where they actually see the same bacteria in different locations and countries because they can go up into the atmosphere and they're really light, and so they travel everywhere. They're down in the Marianas Trench, everywhere. And so bacteria, they get infected with viruses the same way we get infected with viruses. So these viruses are called phage. And bacteria phage They outnumber bacteria about 10 to 1 in the world. So there's even way more phage than there are bacteria. And so this is going on in your body right now as we speak. There's this constant arms race between viruses and bacteria. And the bacteria is the green thing having a bad day, and these pink dots are the phage. And so what they do is they inject their DNA into the cell, and then they kind of steal the cellular machinery of the bacteria, and they make more phage, and then they burst out, killing the cell, and thus exponentially increasing the number of phage. And so what CRISPR is a bacterial immune system. So that's what CRISPR is. The clustered regularly interspaced short palindromic repeats are just a fancy acronym for this immune system. And I won't go into detail about how it all works, because tonight we're more interested in the applications. What are people doing with this? But essentially, it's just a way of bacteria to defend themselves against viruses. The cool thing with that is it's an adaptive immune system. Up to the late 2000s, we knew a lot about immune systems in bacteria, but we didn't realize they had adaptive immune systems. So you and your body right now have adaptive immune systems, right? That's when you get the flu shot, and your body makes antigens and it prepares itself. It has a memory, so when you get infected again, you're armed. So that's what CRISPR is. It's an adaptive immune system. When a phage comes in and infects, CRISPR takes a memory, a snapshot of that infection, and uses it against the phage if it decides to come back. So it's an adaptive immune system for bacteria. And so that's actually what a phage looks like in real life. They look like little moon landing things. So CRISPR in a nutshell. So that's what was discovered in the 2000s, and then up until 2012, when the seminal paper came out from the Dowden lab at Berkeley. They discovered that one of these proteins in the CRISPR system that targets the phage DNA and cuts it. So there's this CRISPR protein called Cas9, and that's what this is. And I'll show a video which helps explain it a bit more. But this Cas9 protein, that's the DNA in blue. So it finds the viral DNA and cuts it. And then once it cuts the viral DNA, the phage can't replicate, it can't destroy the bacteria, it's done. So the researchers in the Dowden lab were like, wow, we could use this as a tool to cut DNA wherever we want. And so I'll get into how we're able to cut wherever we want. So that's the secret, the precision. So now people, how many letters of DNA are there in each one of your cells? Oh, okay, yeah, let me rephrase that. 4, yeah, A, T, C, G. How long or how much DNA is in a human cell? 3 billion base pairs, yeah, exactly. So 3 billion base pairs. So if you were to roll it out, it would be 6 feet long. And each one of your cells, 6 feet of DNA, 3 billion letters. So the idea of having a tool that could target one letter in 3 billion, that's pretty powerful. And so that's why there's so much hype about this. So here's a quick video. And make sure the sound's off. So this is a human cell. And we're going to zoom in. So here's the nucleus. And so we're going into the nucleus is really crowded, right? I just said there's 6 feet of DNA, so it's super crowded. So here's Cas9, that protein. It's floating around looking for a sequence of DNA. And there we go. And then boom. Okay. Oh, no. So let's see. What is happening right here? What is going on right there? So who knows what RNA is? Some people here? Yeah. So RNA is like DNA. The central dogma of biology is DNA makes RNA, which makes protein. So RNA there's a lot of similarities with DNA. And so that's what this orange thing is. This is RNA. And so that's what this is. This is RNA. And so there's a little bit of the RNA that's sticking out of the protein. And RNA and DNA bind one another. So they can form a helix the same way DNA forms a double helix. So RNA and DNA can interact. So you know how A and T forms a bond, G and C forms a bond? So same thing with RNA. And so that's how we program this protein to target one letter in 3 billion letters. So we give it a sequence of RNA. So there's 20 letters that stick out of this protein. And so we say these 20 letters, which are these yellow right here, the scientist says, I want to target this region right here. And so you design the RNA, you give it to Cas9, it floats around the whole genome, and then it recognizes it and unwinds the DNA. So that's what's going on right here. The DNA is unwinding. And then it cuts it. So it's unwound the DNA, and it cuts both strands. And then it floats away. And so now, a researcher, let's say I wanted to introduce a new sequence of DNA. I can just give that to the cell. And the cell has machinery that repairs broken DNA. Because your DNA is being broken all the time. When you go out into the sun, the UV rays, your DNA is always getting broken. So your cells have a lot of really sophisticated machinery to repair broken DNA. So, to recap, in labs now it's really easy to make Cas9. It's really easy to order RNA. So all I have to do is make my Cas9 in the lab, or I can order it. I can go online, type in the letters of RNA I want, and then I can incubate it in a test tube, drop it on a human cell, and then it will go into the nucleus, float around until it finds the sequence that I've programmed it to target, and make a cut. And then I can do whatever I want. I can remove DNA, add DNA, change DNA. So, yes?
So is the is the CRISPR introduced outside the cell or into the cell?
So the question was, is CRISPR introduced outside the cell or into the cell? And so there's different ways to get it into the nucleus, which is ultimately where we want it. Usually people will put little tags on the protein, and there are little tags that the nucleus the cell recognizes, and it engulfs it. Yeah. So you have to sometimes play a few tricks. Another way is to electroshock the cell. So if you shock it, the pores of the cell open up. That's another way for this to get inside. So there's a couple different ways. Yeah.
What separates this from previous generation, I guess, DNA cutting mechanisms?
Yeah.
Restriction enzymes, I think, like, if I remember correctly, like zinc fingers.
Great question. Yeah. So this is not the first protein to ever cut DNA. There's been many, many others, and some of which are even in clinical trials now to treat genetic diseases. The big difference are the thing recognizing the DNA in all previous technologies was the protein itself. And so in order to say, I want this to target this sequence of DNA, I needed to find a way to evolve or change the protein itself, which is really hard. But with this, why it's so powerful and easy is all I have to do is change the RNA, which is really cheap and easy and fast. I can just go online, type in the letters of RNA I want that matches the DNA I want, and it'll come to me in the mail, or I can make it, and then you're good to go. But in previous generations, if I wanted to target a specific sequence of DNA, I needed to find a protein that itself recognized that sequence, which is possible, but it takes about like $10,000 to do that. Whereas with this, you can do it tens of dollars. Yeah. Any other questions? Yeah.
So, follow up to that question. We used to use primer-based gene modification. So you would order primers for $50 a piece, and you would be able to essentially replace the sequence of DNA. How is this different from that?
Yeah. So so for the primer, so I guess for that one, you're doing you're altering DNA that's outside of a cell, right? For this, I've taken DNA, it's outside the cell, I'm adding these primers, and I'm changing the DNA. But this is all done inside a cell. So the primer stuff won't work inside a nucleus, for example, or yeah.
So you skip over all the vector creation of it, you do it in one step. Is that accurate?
Yeah. So for like the type of experiment you're thinking of, One way to deliver Cas9 into a cell rather than just delivering the protein itself would be to deliver the DNA that encodes the protein. And in that case, I would want to use primers, vector design, to design the plasmid I want. It has everything, and then inject that into the cell. But when I'm dealing with the genome of a butterfly or the genome of a human, you need to use Cas9 to go and cut. Yeah. So, one cool video that was taken 2017, so I guess it's already 2019, was actually showing Cas9 in action. So this video, this is actually Cas9, and this is the DNA. And you'll see in a bit that it actually cuts the DNA. There you go. And it cut it. So this is actually what it looks like in real life to see this really cut DNA. So pretty cool. Yeah.
Is it typical to see that kind of spacing between the curves of the DNA actually in the cell like that? That's the amount of spacing between curves of the DNA?
Oh.
Yeah.
I think that's more of an artifact of the experiment.
I see.
Yeah.
Okay.
Good question. Yeah. So let's dive into applications. Oh yeah. Now let me dispute everything I've just said. So what are the 4 misconceptions? And a lot of what I'm going to show you is based on maybe headlines, magazine covers, what's kind of the media saying about it. As you know, everything's context dependent, so let me give you the context of how this all is happening. And usually I end a lot of my talks talking about crops, but today I want to start talking about crops. And so CRISPR-edited crops are GMOs. That's actually a misconception, and we're going to dive into that. So Jennifer says, or a lot of people ask Jennifer, when is CRISPR going to reach the public? When will we have our DNA edited or treat a disease? And her answer usually is, it's going to be through your food. And we'll talk a little bit why isn't every genetic disease cured already? Because it takes a while. But food, the landscape's changing quickly, and people are making a lot of huge advancements in using CRISPR in agriculture. CRISPR is fast, cheap, and easy. I just told you how fast, cheap, and easy it is. Okay, well, let's set the context. Why is that kind of a misconception? And then designer babies are coming. And everyone wants to talk about designer babies. So we'll do that. So previously, these are all the model organisms that maybe some of you have worked with. That's just what everyone in science has worked with for the last 100 years, right? You have your zebrafish, your frogs, your mouse, your fly, your C. elegans, Arabidopsis, yeast. And so science community was kind of confined to these organisms. But now CRISPR, in only 4 or 5 years, has edited over 300 different species of organisms. So in a few months, we're releasing our CRISPR gallery, where you can go online to our website and look at every organism that's been edited, and go to the paper and see why did they edit it. So yeah, the CRISPR has changed the landscape of just basic research. We can alter the DNA in organisms that we could never alter the DNA before. And so let's dive into what is a GMO? Can someone give me a definition? It's a deceivingly hard question considering everything we eat says non-GMO on it. Yeah.
So I guess I'll answer the question by qualifying. If you're in Berkeley or Venice, GMO means completely different versus if you're in science community asking what is a GMO.
Yeah.
My definition of GMO is a genetically modified organism.
Yeah.
You go and write a specific gene. Most things that we consider GMOs are actually selective breeding or not.
Yeah, so it's complete. So GMO itself does actually not have a legal definition. It actually does not mean anything legally. So the misconception of altering DNA in food makes it a GMO. That's a misconception, and I'll show you why. And thus CRISPR-edited crops are GMOs is a misconception. And so I think this is a really important conversation to have tonight, because these rules are changing as we speak. The USDA, the FDA, the EPA are all talking right now, trying to catch up. The technology has outpaced the rule book. essentially. And so why I bring this up is because the ruby red grapefruit, so if you go online to their Wikipedia page and read about it, that was actually, a lot of the ruby red grapefruits you eat are made via mutagenesis. So they shoot x-rays at it and get mutations in the DNA until you get the traits you want. And then you pick that and you sell those. And so, but by definition, that's not a GMO. So those aren't labeled as GMOs. Mutagenesis is not. And then, who knows what rennet is? Do some of you make your own cheese? Yeah, curdling. So rennet, yeah. Rennet. I don't make cheese, I guess. Rennet is most rennet for pretty much all cheese in the U.S. is synthetically made from E. coli and yeast. So it was engineered in yeast. So by definition, then all of that's GMOs. So there's a misconception of what a GMO is. So how does CRISPR fit into this? Great question. And so a lot of these misconceptions, I'm going to zoom into a case study, and we'll go from there to lay the landscape. So this is the white button mushroom, right? How many of you enjoy these? They're quite delicious. Yeah. So what happens with the white button mushroom when you cut it and leave it on your counter for 2 days? Turns brown very quickly, right? Like most produce, apples especially, and mushrooms. So a lot of these vegetables have the same genes in common. So that's my fancy DNA. The polyphenol oxidase. So oxidation, right? Like with rust and whatnot. So oxidation, they turn things brown. And so this particular species has 6 of these genes. And so this research group out of UPenn turned off one of them, used CRISPR, went in, turned off one of those genes, and reduced the levels of this protein down to 30% of what it usually is. And in doing that, now you have a non-browning mushroom. So you can cut it, leave it on your counter for 2 weeks, and it won't turn brown. So why is that not a GMO? So the USDA came out recently saying that this is not a GMO. Actually, it's on the next slide. I won't ruin it. So other examples of where this has been done is the Arctic apple, which is actually being sold in the Midwest right now. Same thing. That was not done with CRISPR. It's done with a RNA interference, if some of you know about that. But similar concept. They targeted the same type of gene, turned it off. Now it won't brown. And then also same thing with this potato. So the USDA has said that they do not currently regulate or have any plans to regulate plants that could otherwise have been developed through traditional breeding techniques. And so how does that tie in with CRISPR? That ties in with CRISPR because imagine we have that mushroom, and we have that gene, and we just want to turn off a single gene. Turning off a single gene can be done in many different ways. It could mean just changing one letter. All right, so genes start and end at a certain spot. So let's just change one letter so that it doesn't start or that it doesn't end. And then, so the USDA says, well, that could easily happen in nature. Letters are always changing all the time whenever you grow anything. And so their argument is that we can't regulate this as a GMO. And so a lot of what scientists have made in the past decade, they're remaking it with CRISPR just out of semantics, right? We've done this with previous technologies. This is what we have come with. We've come with disease-resistant cacao or drought-resistant wheat, but the USDA is saying we need to spend $1 million to prove that it's X, Y, or Z. Now let's just remake it using CRISPR, and now we don't need to spend $1 million to put it through unnecessary regulations. So, semi-controversial. Any thoughts?
So,
Yeah.
so if you put a fluorescent protein in a fish, you are not, that is not a protein that normally occurs within that species of fish.
Yeah, that's definitely
So that's
definitely not. Yeah.
But
yeah.
if you're just messing with the genes that are found in that species, which you could do through selective breeding or through some other technique, that would not be considered genetically modifying that organism.
Yeah, yeah.
So extant genes versus introduced genes?
Exactly. So yeah, one thing is like, let me take a gene from an orange and put it into a banana. There might be benefits to do that, but that doesn't fall under this sentence.
A banana would not normally develop genes
Exactly.
for oranges.
For future questions, should the audience have a mic? What do you think?
I can hear them, and I'll just re-ask them.
Do you think they heard Jeannie's question?
Her question was if you take like a gene from another completely different organism, a fluorescent gene or a gene from a banana, and add it to whatever. So that doesn't fall under this. That's regulated as a GMO. As a GMO. Yeah.
I'm not anti-gene question, but one of the things about turning off that gene in the mushroom so that it doesn't turn brown and let it, turn out for weeks, that it can go bad and you're eating a bad mushroom.
Oh, exactly. I mean, there's a reason why it turns brown, so you don't get sick. Yeah.
So think about why am I doing this?
Mhm.
Is it efficacious?
Yeah. So the big thing with
Repeat the
question was there's often with anything biological there's a reason why it's evolved that way. So for turning brown after 2 weeks, it's probably not good. You shouldn't eat it. So brown is saying it's a red flag. So with the Arctic apples, a big reason why they wanted to do that is because kids get those little Lunchables with cut apples in them, right? Already cut, and so let's have these apples in there so they won't turn brown. You can ship them. It's for pre-cut food and delivered in that. Yeah, and then for this browning in the potato was for a couple of different reasons. It was also doing with disease resistance and whatnot.
So that could be a definition of GMO. That sentence could imply a definition of GMO. Is that the definition that, say, the GMO project uses when they declare something to be non-GMO?
To be 100% honest, the question was, what kind of definition does the non-GMO project use, essentially? And I do not know their definition, only because I've seen their label on like salt containers. And so that, to me, I've seen their label on things that don't have DNA.
I understand.
Yeah, so I don't know
Vodka.
what rules there. They have rules, but I don't know what the rules
Yeah,
are.
and they might make sense.
Yeah, they might make, yeah. But I think that it's gotten to a point where it's marketing and I like everything, but
Yeah, it's definitely marketing.
yeah. Yeah.
If you basically want to know whether a product on the grocery shelf is GMO, look at it, and if it says gluten-free, it's probably GMO, and they stuck a label on it to convince you that it's healthy anyway.
Well, the big thing, so now with the rules changing, there's the scientists are trying to figure out, how can we communicate with the public? It's important that they know what they're eating and how they're eating. So they are going to roll out new labels for biologically engineered, BE labels. And scientists, there are field trials looking at, if we put a QR code on these that go to a place saying this is the gene, this is why it was done. Well, how does that affect consumers' opinion or understanding or whatnot? So they actually did a field trial in Vermont, and it made people feel more comfortable, even if they never actually went to the website to learn about it. They were just happy to know that there's transparency and whatnot. So this is a huge science communication hurdle we're trying to figure out, but I think we'll move on and we can come back to agriculture.
It's a side issue about the mushrooms, and if a mushroom or an apple or potato is engineered or changed so that it doesn't brown, I got the impression that someone was suggesting that it might go bad even though it's not brown. Is that true?
Yeah. There's
I see.
Things that can go wrong with it.
Yeah, definitely. And fungus can start growing that you maybe can't see, but especially if you're leaving at room temp. So crops. So just to be very transparent, at the IGI, we're editing wheat, rice, triticale, maize, soybean, barley, cacao, cassava, sunflower, peanut, broccoli, tomato, tobacco, pepper, and sugar pine. So all for very different reasons. Cassava, because it builds up cyanide. So if you don't cook it correctly, and it's a huge staple crop in a lot of Southeast Asia and Africa, so we're editing it to remove a gene in that pathway, so it doesn't make cyanide anymore. Peanuts, you can imagine, to remove peanut allergies. Broccoli, so there's actually a natural chemical in broccoli that is beneficial for anti-cancer. And so we're just changing that gene to make more of that compound. Rice to be both, well, one person at the IGA made it to be monsoon resistant. So rice, right, it grows in wet paddy fields, but if it's submerged for too many days, it dies. So she edited it so that it can stay submerged for weeks. So these are all kind of the types of projects we're working on. Yeah. One more question.
You worry that you're changing these things and removing something that is in fact essential for the crop.
Yeah, that's always the question was, you know, do you feel like you're changing something and then maybe you're removing something that's essential? And that's the classical question of doing any type of biology that's going to go out into the world. Let's not even get, I haven't even put up here editing mosquitoes to stop malaria. That's a whole different thing. But even though the USDA is not regulating it as a GMO, it's still regulated. Anything that comes out of a lab or a company goes through safety testing.
For example, why did cassava make cyanide?
That's an excellent question. So I was talking with those researchers. The question was, why does cassava make cyanide? And I was talking with the researcher working on this project, and they're still not 100% sure. Maybe it has to do with the pests that eat it, to kill those pests, and they're still exploring that. But the reason why they're aggressively pursuing this project is because it only makes cyanide when it's not prepared correctly. And right now, with a lot of droughts in Africa, people are unable to prepare it correctly due to the type of cassava they're using or the lack of water and whatnot. So it's just this cost-benefit playing. So I'm going to move on real quick, and we'll come back to agriculture. So misconception, CRISPR is fast, easy, and cheap. And so you see this stock photo all the time of the scientists cutting DNA outside of the cell. It's fast, easy. So you see this every week all the time because we're always tweaking this protein to make it more accurate. We're even discovering more proteins like this. And it's just becoming easier and faster. But let's put that into context. So not in the context that, Joe Smith can do it in his garage. This isn't like computers where you can go into your garage and build a computer. It's a little different. You need equipment. And so let's look through the lens of a project we're working on at the IGI at Berkeley in collaboration with Oakland Children's Hospital. And so what disease is this? Sickle cell. Great. So sickle cell disease. So it technically is a rare disease, but it infects a lot of people. And so how are we pursuing this? We're pursuing this because, or where do your red blood cells come from? Bone marrow. Wow. Okay. You guys are on it. That comes from your bone marrow. I didn't learn that till grad school. So your bone marrow. So what they're doing is a bone marrow transplant is a common procedure for various reasons. So what they're doing is you remove the bone marrow, you apply CRISPR to those cells outside of the body, so in a hood, and then you make sure it worked, and then you reintroduce it back into the body. So this is good for a lot of reasons. One, because it's your own cells, so hopefully you don't have an immune response. And also, you don't need 100% efficiency. You need possibly as low as 5% of the cells to be correctly fixed. And that's enough to lubricate the blood and carry the necessary amount of oxygen. And so that's what we're pursuing. Some other labs across the world are doing slightly different approaches for this. But this is fast, easy, and cheap. I explained it in 2 sentences, so it sounds fast, easy, and cheap, but it's still very, very difficult. Treating genetic diseases in general are very difficult. It's very expensive. So this was just approved, this drug, to treat kind of the bubble boy disease. And it's a $700,000 drug. one-time treatment. Another treatment was just approved to treat a genetic disease, and it's a $4 million for one injection. And so these are really, really, really expensive procedures. And so one thing at the IGI we're doing is working with insurance companies and lawmakers, because that's where the price comes down, is from that realm. Like how can you regulate this so that it's still safe, but that it can not be $700,000. And so clinical trials are slow. So we get a lot of emails from people across the world saying, can you treat this disease or that disease? And the answer is possibly, but clinical trials, you need X amount of just basic scientific data before you can even apply for the paperwork. So we have a lot of great data, but we're still a year away from the FDA saying, okay, you're ready to try this on a human in Oakland. So clinical trials could take anywhere from 5 to 10 years. And then you still need expertise. And so I'll just jump to this guy who lives in Oakland, Josiah. He injected himself with CRISPR. What do you think is going to happen? I mean, it's not even going to make it into the cell. You're just going to get an infection from the needle. And so there are biohackers out there trying to do this. It's not going to work. Yeah, but that doesn't mean we don't need to think about people trying it on themselves. So I went to a biosecurity conference in Germany where people from a lot of nations came, and the whole point was just to brainstorm, how might people try this for nefarious reasons. And so we brainstormed, and how can you try to stop these people before they might try to do something wrong. Yeah.
So going back to your sickle anemia example, human bodies are made up of millions of cells, and every one of those cells has DNA. And if you are putting in a new, you're putting in cells into bone marrow, don't you have to do that often to change that DNA change? Because they don't last that long. It's just the body that's making the proto red blood cells.
Yeah.
The erythrocytes. How do you really have that going on?
Yeah, so the question was, in the context of sickle cell, do we need to replace these edited cells? And the question for that's no, because those are the stem cells. And so so they stay. So a lot of these gene editing experiments are done on cells that don't regenerate and you have to replace. So there was recently a clinical trial approved that's for blindness, genetic blindness. So your eyes, those cells where they're trying to target don't switch over. At the Institute, we have someone working on Huntington's. And so your brain is another example where it doesn't turn over. Those brain cells don't change. Yeah. So you want to target stem cells.
Sorry, you might have asked me this already, but I'm still trying to, envision how this gets into an organ. Does it just go through the cells? Or if you're saying, for example, a human, or if you're talking about mushrooms, where is this introduced at? Is it introduced at a single stage, or is it allowed to work when something's already differentiating?
Yeah, great question. So the question was, at what point in either the lifespan of the crop do we do this, introduce a Cas9? At what point, in the human? And so it's context dependent, but let me maybe give 3 examples. So for one, with the crop, crops have embryos also. And so you'll take the embryo of the plant and edit it. So that's for the crop. For a human, a couple different things. For the designer babies, you'll edit the embryo. But for a lot of the experiments that are approved now or that will start in the next 3 years, you're editing somatic cells, which is anything that's not a reproductive cell. And so, but the question of delivery is very important. And that's where a lot of money and time is going right now. How do we get this protein right here to where it needs to go? And so one question with sickle cells, okay, let's remove the cells outside of the body. Remove the stem cells and we'll edit and we'll do all that and we'll put it back in. But that doesn't work for questions like the brain. You can't remove the brain, edit it, put it back in. So one thing to test their procedures on mice, they're actually drilling a hole in the mouse brain, injecting it. So that is actually a way you could do it in a human. But they're finding ways to just coat this protein in a way where you can inject it in the blood and it knows to go to the brain. But your brain has a blood-brain barrier, which is very hard to penetrate. So every organ in your body, there's another issue to overcome, but people are trying to figure out how can we deliver this to the only cells we want to edit. Yeah. So another one is muscular dystrophy is on the horizon. There was some really interesting dog studies where they cured the dogs of muscular dystrophy. In that one, you can do a direct injection to the muscle. Yeah.
So you can either inject it or the technology is still involved, so they need some kind of encapsulated thing that's the cell's going to break out.
Yeah, so you can either inject the protein itself and add tags so the cell recognizes it, or AAV viruses. Some of you might know that. You can use viral capsids, and viruses are built to infect your cells. So inside the virus, you can put what you want to get into the cell. So, designer babies. We'll end on a good note. So these were a lot of the magazine covers from when CRISPR first became popular. And so you can see why this captured the public's attention, where you have a lot of these, I guess, white babies with blue eyes. And so why are or aren't designer babies coming? Now, if you ask some people, they say we've been designing all along based on who you choose to marry, blah, 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 blah. You choose to pass on your traits. Okay, no. But PreImplantation genetic diagnosis, has anyone heard of this phrase? Some people? So PreImplantation genetic diagnosis is a common option in a lot of fertility clinics. It's just a way of not passing down, people often use it as a way of not passing down a negative trait. So let's say you and your spouse maybe both have a disease, but not on both chromosomes. It gets complicated with the math because you have 2 copies of each chromosome. But in a lot of diseases, you have, say, a 50% chance of passing down that disease. So what PreImplantation genetic diagnosis you can do is, okay, let's take the sperm and the egg outside and let's fertilize multiple eggs. And then once they reach like a 6-cell stage, we take one cell and we look at the DNA and we pick the one that doesn't have that genetic disease and implant that. So people, and I think it's a valid argument to say that, we've been, this has been around for decades, so we've been artificially choosing, which genes we do or don't want to pass on, so via this method. Now with this though, you're limited, right? You're limited to only the genes that the mother or father have. I can't take a gene from someone found in another country and introduce it, because I'm only limited to what is being passed down from between the father and mother. So, any questions on this? No. So, and then another correction to the misconception is we can only design so much. So a lot of people will ask, when can we have a super intelligent person? And my response is, we don't know what intelligence is. How do you encode intelligence? It's not. So a lot of things that we can approach are monogenic. So they're caused by one gene, right? Sickle cell is a 1-letter change in one gene. But a lot of our traits are polygenic, right? They're accumulation of a lot of genes. And so also unknown side effects. So I'll give the example after this slide of what they did in China in editing the babies, and there are unknown side effects, obviously, to altering your DNA. Now, sometimes you weigh that. We removed a very deadly gene mutation, and I will accept maybe a lesser of 2 evils, but there's also unknown side effects. Done at an embryo. So I was saying before, to do this, you need to go to a fertility clinic, and a lot of them are not that well regulated, and so people who are worried about this getting out of control have their eyes on fertility clinics. It's expensive. So all of this requires in vitro fertilization, which is not cheap and is often not covered by insurance in certain instances. So when people are worried about, some of us at the Institute are writing a paper now on, people are becoming more and more okay with editing embryos. If you've looked at polls over the last 3 years, but I don't think people understand that that's only going to be for rich people. Right? So when you put it in the context of that, it becomes a lot more cringeworthy. Right? So none of this is cheap. Right? It requires sequencing the embryo DNA. It requires in vitro fertilization. It requires a lot of stuff that only wealthy people can afford. And so that's when you get into this whole only the wealthy people will possibly benefit. Yeah. Yeah, so the question was, for this pre-implantation genetic diagnosis, or even with CRISPR, if we remove a gene, or we choose an embryo that doesn't have a gene, yeah, it's forever outside the gene pool. That person's baby's future offspring will not have that. And so that's why editing embryos is, there are more different ethical questions because the idea of consent. So your future generation, and then we don't know enough basic biology to do a lot of this. We just don't know baldness. I mean, I don't think that's a single gene, or else, I mean, we would fix that. Yeah.
So going back to the sickle cell anemia, one letter. Can you change that one letter and that child no longer has sickle cell anemia? That's a good thing. And you probably do not affect any other gene. But if you're talking about CRISPR taking out a whole set of letters, then you're talking about changing possibly the epigenetics of that particular organism, or the rereading of it in a different way. And
Great question. So, kind of going back to the agriculture, right? If we change too much of the human DNA, do we know what's going to happen? And so, right now in the regulations of what they're allowing and not allowing, kind of addresses that. So, what they say is, if you have a disease or a mutation, you can change that to what the normal state is. So, so that sets your boundary. Let's take what is mutated here and make it look like what the rest of the population looks like. And then you can assume that you won't have, yeah.
As a little comment on some of the questions, not all mutations are adaptive. Not all parts of the genotype are adaptive. Because natural selection is only one of these 5 mechanisms of evolution. So to say what is it for is not always an appropriate question. Okay, it may be for nothing. You may have the question, if you could, if you can replace an ender in a zygote, that would be passed on to future generations. And that would be reason enough. I'm trying to stay away from the designer baby thing, but something that would be needed. Discuss the difficulty of doing CRISPR in zygotes.
Difficulty doing, I don't think that actual doing, so the difficulty of doing CRISPR in zygotes, I don't think that the actual biology or mechanics or procedure of it's any different. Now the ethics, or we can talk offline about the subtleties, differences of that. But I think this is an interesting case study. So who saw this headline? A couple people? A lot of people? Everyone? Okay. So yeah, so this was very surprising. We had a very busy week at the end of November. Because a couple of days after this, they were having the second international summit on genome editing. So all the world's top genome editors, as well as a ton of journalists and lawmakers, were convening in Hong Kong. And so this happened a couple of days before, and this guy was actually scheduled to speak. So it was a very fascinating couple days. So what did he do? So what he did is he wanted to make HIV-resistant babies. And so a small percentage of people of Northern European population have a natural mutation that makes them HIV resistant. So it's a gene called CCR5, and there's a natural mutation that prevents you from getting infected. And so he said, okay, let me introduce that naturally occurring mutation into these babies. And so what he did was he took this Cas9 protein, the egg, and the sperm, and added those three at once, and then did the edit. And then he looked in and see if it worked. And he hasn't published any papers, but based on some preliminary stuff, it doesn't actually look like some of it worked. So there are many ethical things wrong with it, but the fact that he still implanted it is not good. And then he got two girls. So based on the science, are they HIV resistant? I don't know, but it raises the question of, is preventing something, is that morally acceptable? So it's not quite an enhancement, but they just can't get HIV anymore, and it's a naturally occurring mutation in people in Northern Europe. So it's an interesting case study of what should be allowed, what should not be allowed. Yeah?
Do we know if the two girls are actually have the gene that was edited? Did he confirm that part of it?
So he presented slides at this Hong Kong summit, and based on those slides, he made changes that are kind of close to what naturally occurs in people, but not exactly. So, short answer, we don't know. You would have to duplicate that in a mouse or in a human cell and infect it. And so, yeah, but right now, he's under investigation in China because this is illegal in China. And yeah, so, but the Pandora's box has been opened. We have CRISPR edited people. Yeah, so 8:30 on the dot. Drink some wine.
Maybe you have some more questions?
Yeah.
You said that you could theoretically stop blindness, right? So, like, could you make it? Could you make it happen? Like, could you make blind babies?
Could you make blind?
Like morality aside, like, could you have the kids come out blind?
Yeah, definitely. Yeah.
Free student.
Yeah.
Why did he mix all three? Why did he mix all three of them together? Did they need to have both the sperm and the egg take up this protein, or?
Yeah. So the question was, why did he mix all three at once? It's a race. So you don't want the one cell to go into 2, then to 4, then to 8. You want the edit to happen at the one cell stage. Because also what they saw based on the quick slides he showed at Hong Kong is that one of those girls is a mosaic. So you know what a mosaic is, right? Tiling. And so what that means is that not every cell in her body was edited. So that means that the CRISPR didn't make the edit before the cell divided. So that also means that that girl is probably not HIV resistant.
How are advancements in computer analysis, or how are advancements in computer science and maybe neural net learning affecting developments in this area?
So, question, how are advancements in neural net and computing? That's a great question. I don't know. Yeah, I don't know. There's CRISPR blockchain. I don't know. You just throw buzzwords together. There's AI. I don't know. Yeah.
Way back toward the first of your talk, you showed CRISPR cutting.
Can you use the microphone?
And then you showed a different length of base pairs that you got from somewhere, some DNA that was inserted, and you said, well, it just knows to do that and repair itself. Where did that length come from? What makes it special that that's what's going to be used in the repair?
Yeah. So he's referring to that video I showed where the sequence of DNA got inserted. That was just a hand-wavy cartoon showing we can insert DNA. The long story is, if we do want to insert DNA, how do we know where to tell it to go? And so imagine, boom. Okay, so imagine you make a cut in this nice little rainbow. We go over here, and you give the DNA you want inserted, and you say, okay, these letters will match here, these letters will match here, and then the purple goes in. So on the ends of what you want to insert, you match that to the ends. And so that's how you tell it where to go.
So there's some replication of pairs at the intersections, and that's used to match, and then the duplicates are discarded as it gets merged in.
Yeah, they don't. Yeah.
Where do you get those things that you're inserting? Are you using CRISPR to chop something else?
Oh, this? You order that. You can order DNA.
And build it.
Yeah.
Now, technically, if you inserted a gene that was either taken from another organism or sequenced, you actually would be creating a GMO at that point if you cut the DNA and then you inserted a foreign gene in the middle of it and all went together and reproduced from that on.
Yeah.
That's GMO.
So if this DNA was like an orange, from an orange, and this was from an apple, and that would be a GMO. But this actually wouldn't be a GMO. So what happens, this is actually quite hard. The efficiency is very low. This is another way that our cells repair DNA. And so what happens is you make the cut, and the cell says, oh no, there's a cut, I need to quickly repair it. And when it does that, it usually makes a mistake. It will remove a letter, add 2 letters, something. And so with DNA is read in blocks of 3 letters, right? So that makes an amino acid in blocks of 3 letters. So if you add one, you've thrown off the whole DNA in a way. So this is a way that scientists turn off genes, is we just make the cut and we just let the cell try to repair it, and in the process, the gene is turned off. And so if we did that in an orange to turn off a gene, then that wouldn't be a GMO. Because evolution could have easily have just knocked that letter out or added 2 letters. Yeah.
So you say evolution could easily do that. Can it? Really?
Yeah, so it's a numbers.
The odds of a particular gene being edited out in a particular way are incredibly low.
Yeah, so it depends what...
What's the question?
The question was, when I say nonchalantly, evolution could easily do that. So good question. So let me paint a picture of that mushroom, where I want to turn off that one. There's 6 of those oxidase genes. I want to turn off one. So there's different ways to turn off a gene. I could just remove one letter that says don't start reading that gene.
Right.
And changes in one letter happen all the time. So if you were to measure every cell in that plant, there's the numbers game, right? But that would not be the most efficient edit to make. If we really wanted to turn off the gene, maybe we would remove a larger sequence of letters. And in that regard, maybe over time that could have happened, but that's less likely. So yeah, you want to make not a huge change, but like a robust change, so that the gene is sure to be turned off. Yeah. So to answer your question, a lot of times to even know which gene to edit, they find a certain crop. You'll see a crop in the field that happened to withstand a drought or withstand an infection, and you go in and you study. So the hardest part with all those crops that I listed that we're working on is even which gene do we even go after. And so going into the fields and saying, oh, that crop withstanded this locust infestation or this beetle or whatever, why did that happen? What's special about that crop? And so let me take that and then mimic that feature it had. So a lot of times they're not recreating a new trait, they're just identifying traits from other ones and then adding that to the species that you're interested in.
So going to the sickle cell or single gene diseases, showing promise that you'll be able to treat this. For things that are more advanced or more complicated, such as cancer, maybe there's dozens of genes involved. How close do you think we would be to having maybe a cocktail delivered for potential treatments in the future? Is it not efficient enough for things like that to be able to happen?
Yeah.
I just want to get your thoughts on that.
Good question. So cancer, what they're doing is they're not editing the human cells that have the mutation. they're editing our immune cells. So what they're doing is, and this is already in clinical trials, where let's take our immune cell out of the body, edit it, and tell it to recognize and aggressively attack the cancer cell, and then put that back in. So a roundabout way, yeah, if you're trying to target a ton of different mutations, that's, yeah, very difficult. But play some games. Tell the immune cell, make one edit that makes it really aggressive at attacking virus or the cancer and put it back in.
There's also research in another direction with CRISPR on proto oncogenes, so that you approach from the cancer cell itself, from the stem cell, it becomes a cancer cell. A number of fronts it's being used in.
Yeah.
Maybe I can ask a question here. Over the last few days, there was a conference here in the Bay Area of industry and science, industrial specialists and scientists concerning what is called clean meat, production of cellular agriculture and cultured meat, meat grown in vitro. And I understand that some of the motivation for that is those who are concerned about the suffering of animals in factory farms and even just in the process of slaughter. We eliminate those animals with cultured meat. You told me in private that we know the gene that will make animals incapable of, some animals perhaps, of feeling pain. Is this another road to agriculture, to animal production that will minimize suffering?
So, yeah, exactly. So someone at Davis also changed the gene so that a cow didn't grow horns, right? So you know dehorning is very painful for cows, so why not just have the cows not have horns to begin with? So that's another type of thing where it's, it's the lesser of two evils. Yeah, so I guess that depends on, well that's a tough one, yeah.
What's the evil in removing the horns?
Oh, well,
genetically removing the horns.
I think if you ask a lot of scientists, they would say there's no evil, it's fine, it's a healthy cow. But whether or not engineering a cow not to feel pain, that's like, I don't know, cutting hairs, but yeah, that's a tough one. But you could do it. Yeah, because there are humans out there who have mutations who can't feel pain.
And they end up getting bruised.
Yeah, so they actually, life expectancy is a lot lower.
Pain has to function.
Yeah.
Unfortunately.
Yeah.
Pain is an adaptive mechanism.
Yeah.
Pain tells you that there's something wrong.
Yeah.
You eliminate pain, and a cow can't tell you that she's in trouble if she doesn't feel the pain. That creates the message that there's trouble.
Yeah.
And so the cow may act perfectly normally and be about to die from something that's killing her. Yeah, another example. So be careful what you ask for.
Oh yeah. No, I'm not asking for that. But yeah, that's a thing you could do. Yeah. So actually, I was talking with a researcher who said, okay, in what instance could not feeling pain, be beneficial for a human? And they were saying, in chemotherapy, right? A lot of times you can't attack cancer because you reach a point where you can't withstand the pain anymore. So there are actually people looking at epigenetic editing. So also using Cas9. I won't go into the epigenome, but it's a way of altering the DNA without permanently altering it. So let me turn off the pain gene just for while you're doing chemo, and then we'll turn it back on. So those are like instances where, okay, maybe that could be useful.